Hi, good evening friends and my dear loving students. Once again I am coming with the second video for the benefit of student community and for the benefit of myself, so that I can engage myself in a very positive manner. And maybe if this video helps you, maybe this portion today what I am going to cover starts maybe between 7th and 10th, I do not know. But whoever it is, I just want to go the reverse way. Once you learn about biology about the plants, the first thing you know that it is divided between angiosperms and gymnosperms. So the angiosperms means the plants that bear the fruit are called angiosperms. So again angiosperms, we divide them further. So I'm not getting into classification because today my focus is only on to let you know how to identify whether it is a dicot plant or a monocot plant. So at least a little introduction I am going to give you about what is dicot and what is monocot. So by word itself, mono means one, di means two. So naturally cotyledon, cot is a short form for cotyledon, that means seed. So monocot means the single cotyledon, single seed, the seed that cannot be broken into two pieces. Whereas the dicot means the seed that can be easily broken into two equal forms. So very, very simple. That is the simple definition and the correct definition also. So most of the common examples that you have got everyday life, you see our groundnut, famous children's favorite, it breaks into two equals. So if you keep on looking into pulses in your kitchen, you will get plenty of examples for dicot. So during this lockdown time, you can identify in your kitchen, and then easily you can bring out at least 10 to 15 dicot. But what about monocots? So monocots certainly are very few, but the number is more. But examples you get very few. In kitchen it is difficult to get it. But I will just tell you simple examples is rice, wheat, our onion. If you grow onion, you will come to know that's another best example of a monocot. Maize. So these are some of the few examples that you can have it. So today I am going from bottom to top. So theoretically in your textbook you will learn how to identify the dicot and monocot, and you will have a tabular form, and then you will also do the get the marks also, maybe out of out also. But that's not my interest. My interest is to suppose I give you a flower, or I give you a leaf, I give you a root, or I give you anything without even telling you the name of the object that I give you. You should reach a level where you can identify it. That is the ultimate learning, isn't it? So today I'm just beginning with a very simple thing. I will keep showing you, keep watching, then we'll come back to the same position later on. See, I am under this beautiful plant, and you can watch this. Just started to flower, and you can watch this fresh leaf that is fine here. Okay, you can see this leaf, we call this as venation. You can see the central rib, and then parallel to that central rib you can see the veins. Okay, these are the, then again we can go further down. So I can show you your famous another money plant here. The money plant doesn't bear the flowers, so I just cannot show any flowers. I can only show you a big leaf. Again a midrib, and then veins that are coming from that, and then it goes on like that. The structure of a leaf also I am not looking into it. Then here I can show you another different type of leaf. This is you can see the difference. There is a midrib, and if you see closely, instead of from midrib, here the veins are going parallel. Isn't it? Can you observe? So this is the difference very visible in this leaf structure and the other leaves that I have shown you. So you keep this in the mind, and then that is between the, I showed you a difference with the venation. And again, we have plenty of leaves here. The shapes also are different. Here also there is another beautiful leaf, you can see. And here the veins are all distributed. Venation pattern is different in this, but it is the same. Again, instead of only one midrib, they are from the same origin, they are all getting distributed. I am not going into depth of venation. That is called, the structure of veins is called venation. And here also, again one more plant, you can easily see the leaf pattern. The lines very visibly from the midrib, instead of going like this, they are all going straight. So such venations are called parallel venation. This is another beautiful leaf, our haldi, turmeric. See, very beautifully you can see the central midrib, and then all the veins are starting from the midrib. And then there is an intricate further to that division. So this such a venation is called reticulate venation. Reticulate and parallel venations. There are two types of venations. Venations means the structure of vein, and then the variety is reticulate and parallel venation. So any number of plants that I show you, you will keep finding only the difference in the venation as I am telling you. So how many types of venation we found right now? One is the parallel venation like this, very visibly, all are coming like this. Parallel venation, and you can see here beautifully is a reticulate venation, you can see. They are all networked. Whether the leaf is small or big is not an issue. The reticulate venation, you can see from here all going further to that, you can see the area boundaries and all. So this is another, this is called reticulate venation, this is called parallel venation. I will tell you then. Again here we have got the same, you can see the reticulate venation leaf, and I can show you the flowerlets. So here I will just take one from that. How many petals are there, children, in this? You can see there are 4 petals, isn't it? So anything you see, any small one, which wherever there is reticulate venation, I can show you 4. What are they called? Petals. So in generally, reticulate venation flowers will have either 4 or 5 petals in multiplication. It will, the number you can study with any number. See, I can show you another plants also. You can count 1, 2, 3, 4, 5. 5 petals. Again, I have another flower here. Same. 1, 2, 3, 4, 5. Then another pretty one. I love this plant because it gives 365 days flowering one. Lovely small flowers, but 1, 2, 3, 4, 5. So either it is a total bunch or it is this thing, all either they will be in multiples of 5 or 4. This is in respect to reticulate venation plants. Now, unfortunately today I have beautiful monocot flowers also, but now right now I don't have, they are not in blooming state. So here I brought one, you can closely see. This is the one, it's already torn. And then the second, third. There are only 3 petals in this. And its leaf also, if I have brought it, I can show you. Okay. This is, this flower's leaf is again, see, this 5 petals one has reticulate venation. This is also, this one's also has reticulate 5 petals and reticulate venation. 5 petals and reticulate venation. The other one I showed you 4 and reticulate venation. This is the one which has only 3, but it is I'm not able to give you as a good example because today none of them have bloomed. So I can show you another example also here. Another lovely creeper. See, you can easily count 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. So 5 petals, and you can see the venation. It is a reticulate venation. Yes or not? Isn't it? So there are umpteen number of ones like that. One more here is there, cactus. That is also reticulate venation, and also same. This is actually it's a different name we have to give. Totally 4 are there, but 2 to combine. That's another beautiful pattern. You can see these 2 are combined. These 2 are combined. There is no separation between them. This is also reticulate venation with 4 petals. So this is the basic structure that I am giving you, the difference between flower pattern and the leaf pattern. So what I need to give you is with the root system. So I should children, just now till now we spoke about leaf structure, then flower, particularly petal structure and number. Now I am coming to the roots. I will show you 2 examples. You can see this closely. By now you should be able to understand that this leaf has a reticulate venation, isn't it? Then observe the root also. So how is this? There is one single one that has come down, and then from the single, like how from the midrib the ribs, all the veins will come. Similarly, from the single mother root, the side roots have come. Can you observe? So this is one type of a plant, reticulate venation with taproot system, where the mother root will come, from mother root all the branches will come. Then I am having another example here. I was telling you, onion is another beautiful example for monocot. See here. You can see this leaf with, by now you should know the term, parallel venation, and here there is a bulb. And you can see, do you find any mother root here? No. All the roots are started coming out from the same spot. So this type of root system is called fibrous root system. There is no mother root which is coming straight down. Everything from one place only they have come. So this system is called fibrous root system, and then this is called taproot system. This is the basic difference between the leaf structure, the root structure, and the petals and the pattern. Another interesting point I am going to tell you. Luckily today, very accidentally, maybe my intention is good, I just now found one. You can see this. All of them, they are going to wither right now. So you can see the taproot system down, and then just the plant grew up, and epi, you can see that the cotyledons have come open. Now the leaves have started. So this is how another good way of identifying. So because 2 have come on the sides, cotyledons have grown like this. They appear to look like they are leaves, but they are not leaves. So they wither out, and then the real plant will shoot up. So this is a definite dicot, because there are 2 dicot plant. I don't know which one it is, but by seeing the taproot system, by seeing the 2 cotyledons on either side, I have identified that it is a dicot plant. For in case of monocot, they will be, the only single cotyledon will be there. That cotyledon will open up under the soil. It will not open up above the soil. So now I have given you, basically, I just again repeat, I have given you, I have got only one example for monocot. I do have them in my garden. but today they have not blossomed, so I am unable to tell you. So we will go back to our original place we started. I have got different patterns, leaves also so beautiful here, and we started here. So again I repeat, leaf structure, reticulate venation, whereas here the leaf structure, parallel venation, and roots are in my hand only, taproot system and fibrous root system. So based on these things, now you should be able to identify. I have given you a clue here in fact. That is, there are two cotyledons opened up and there is a taproot system. And there is one more internally also, which I will not be able to help you out, because that's based on the vascular bundles. When we open up and see under the microscope, then the vascular bundles will be evenly distributed in the center for a dicot and unevenly distributed across. Under the microscope you can very visibly see. So that is with respect to internal structure. So now I have just to sum up, I hope the first point with the root system, what is the difference? If it has a fibrous roots, it is monocot. If it has a tap root, it is a dicot. Coming to leaf structure, if it has parallel venation, it is a monocot. If it has reticulate venation, it's a dicot. Coming to the flower part, almost all the dicot flowers will have 5 petals or multiples of 5 or 4. In some cases they are 4 also. I showed you one example there. Whereas with respect to monocot flowers, all of them will be 3 or 6 or 9 multiples of 3. So if you are given an example of the flower diagram also, you should be able to identify. If you are given the root structure also, you should be able to identify. If you are given simply leaf also, you should be able to identify. And you should be able to see with the venation also. So these 4 factors externally will be able to tell you, even if you are not given the name of the plant also, which is dicot, which is monocot. So this very simple attempt to practically tell you how to identify. Totally 5 differences are there. The other one is a vascular bundles, arrangement of vascular bundles, which is internal structure. So if it is a monocot, they are not in order. They will be either at the edge or in the middle somewhere. Whereas in case of dicot, vascular bundles will be very visibly, centrally, circular form, they are located. So that is an internal structure wise. These 4 are enough in the exam as a hot question. If you are not given the name of the plant, you are either simply given root or a leaf or a flower or a venation, you should be able to identify with this event. So thank you. God bless you all. Enjoy the vacation. Stay home, stay safe, and don't think corona also is good for us. So caronaa in Hindi, kuch caronaa. So I am doing this. कुछ करोना बोल के मैं कर रहा हूँ ये आपके लिए. धन्यवाद. Thank you. Bye bye.